Glofitamab is a CD20/CD3 bispecific antibody, which binds CD3 on the surface of T cells and CD20 on the surface of B cells. And it has a unique 2-to-1 conformation. So it binds 2 CD20s to 1 CD3. And in a publication early this year, late last year, in the New England Journal of Medicine, we showed that glofitamab induces complete remissions in 40% of patients with relapsed diffuse large B-cell lymphoma and at least 2 prior lines of therapy, and that the main adverse event from glofitamab is cytokine release syndrome, which is a common side effect that is an on-target effect from activating T cells. Glofitamab is an intravenous agent, and it's given after a single dose of obinutuzumab, and then a week later, we introduce glofitamab. Now, this clinical trial that we're talking about today, which has a code NP40126, is a trial exploring the combination of glofitamab with Pola-R-CHP. And the reason this is important is because we know that Pola-R-CHP is probably one of the most important advances in the frontline treatment of diffuse large B-cell lymphoma, because we have a large randomized study, the Pola-R-CHP study, which shows a progression-free survival advantage from the supplementation of polatuzumab for vincristine in patients with IPI-2 and above disease. That has not yet translated into an overall survival advantage, but even that PFS advantage has led to approvals of polatuzumab vedotin as a part of frontline therapy globally in many regions. And so what we want to do is now deal with the area of need, which is the group of patients who don't achieve a complete remission from that Pola-R-CHP combination, by taking this new, very active agent, glofitamab, and adding that to the combination. In a way, it's a pragmatic combination, and this study is part of building a data set that justifies future clinical trials, we hope, in evaluating the addition of glofitamab to frontline therapy for diffuse large B-cell lymphoma. So in this clinical trial, the schedule is fairly straightforward. Patients receive a single cycle of Pola-R-CHP, followed by a second cycle of Pola-R-CHP, and we add glofitamab at its first step-up dose on day 8, followed by a further step-up dose on day 15, and then the final step-up dose on cycle 3, day 8. We don't use obinutuzumab in this combination because rituximab is used, and that probably has a similar effect in terms of abrogating cytokine release syndrome. We included patients who were aged greater than the age of 18, had an ECOG performance status of 0 to 3, and an IPI score of 2 to 5, and a diagnosis of diffuse large B-cell lymphoma that had not yet been treated. So far, we have preliminary data that we're presenting. So a cohort of 24 patients who've been exposed to treatment. And what we saw was a really striking signal that the combination is safe. So the rate of cytokine release syndrome that we saw in this population was 12.5%. Only 3 of 24 patients who were evaluable for safety developed cytokine release syndrome. And we saw no examples of ICANS, that neurologic syndrome that occurs after T-cell activating treatment. So that shows the combination of chemotherapy and glofitamab is deliverable. We had full dose intensity of the chemotherapy, polatuzumab, glofitamab combination. And we had acceptable rates of neutropenia and fever after treatment. Patients got through treatment on time. We had 17 patients who were evaluable for efficacy, and the complete remission rate was 76.5%. The vast majority of patients had high IPI disease on this trial. The overall response rate was 100%. What we saw was that the best response was not associated with the IPI. So patients who had IPI of 4, for example, there were 5 patients who had an IPI of 4, they had a complete remission rate of 80%, partial response rate of 20%, whereas patients who had a IPI that was low also had a complete remission rate of 83% and a partial response rate of 16.7%. So we're seeing a good rate of responses from this combination. Now, obviously, we need more durability and we need more patients to understand overall what the performance of this combination is. But this preliminary data suggests that there is a justification for exploring the combination further. Well, we're presenting this data relatively early, but this is a good number of patients to understand that you can combine Glofitamab with Pola-R-CHP in a way that does not seem to compromise the chemotherapy polatuzumab backbone. Frontline randomized clinical trials of adding a new agent to an R-CHOP backbone have often failed due to loss of intensity of the R-CHOP backbone. With this data, we've shown that we can deliver Glofitamab, and there doesn't appear to be a loss of intensity of the R-Pola-CHP backbone. So that suggests that a randomized study would be feasible from a deliverability of the backbone point of view. And so the next step is not just to show that the combination is safe, but also to show that it is more efficacious than the standard of care, which is Pola-R-CHP. That requires a randomized clinical trial, and obviously more thought and consideration needs to go into that step. But that would be the next step in terms of developing this particular combination regimen.